Hi everyone, today we'll be talking about revolutionizing plastic recycling using PETACE and METACE. My name is Apoorva Agarwal and I'm joined here by my teammates.
I'm Tommy Bradford.
I'm Drew Lazaro.
I'm Sarah Scroggs.
I'm Michael Zhang.
So talking a little bit about the problem that we're trying to tackle. So since the mid-1900s, over 6 billion tons of plastic have been produced, and not even a tenth of that has actually been recycled. And as we all know, plastic pollution has a lot of harmful effects on the environment at large, harmful effects on wildlife and on people. And there are a couple of reasons that recycling hasn't been as effective as we want it to be. One of those reasons is that plastic products are extremely cheap to manufacture from raw materials. And secondly, a lot of the times the recycled plastic is not as strong as plastic manufactured from virgin grade or raw materials. And that means that companies don't really have much of a financial incentive yet to use recycled plastic. Our solution to this problem is to use PETACE and METACE, which are recently discovered enzymes, to break down the common plastic PET into virgin grade monomers that we can then sell to plastic manufacturers. After we scale up from the bench scale, we're hoping to first meet the benchmark of treating 75% of the plastic in Washington, D.C.'s landfills. And after that, we are hoping to scale up to treating 10% of the world's consumed plastic. And another benefit of this process is that because it will be on an international scale, it will also cut down on the need for international export of recyclables. A bit of a technical summary of our process is that we'll be producing PETACE and METACE, which we will refer to as PAM, using recombinant E. coli. We will purify it, and then we are going to use it to break down the PET, and then we will purify the monomers for sale to manufacturers. For visual representation, so we obtain the PET from the landfills, we will heat it and cool it so that it's amorphous. We'll be producing the PETACE and METACE separately, purifying it and combining it with this amorphous PET to be sent into a treatment reactor. The effluent will then be purified. The main products will be ethylene glycol and terephthalic acid for sale to plastic manufacturers, and the main waste will be BHET and MHET.
So a little bit about the biology behind our process. Ideonella sicyensis is a bacteria that was discovered in Japan in 2016. It secretes PETACE and METACE naturally. PETACE and METACE are both enzymes that are comprised of alpha-beta hydrolase folds and have catalytic triads. So for the process, PETACE and METACE work together to break down PET into constituent monomers. PETACE first binds to the PET and cleaves the ester bond, producing bis-2-hydroxyethyl terephthalate, or BHET, terephthalic acid, and mono-2-hydroxyethyl terephthalate, or MHET. METACE then hydrolyzes the MHET to produce additional terephthalic acid and ethylene glycol. Research suggests that the optimal conditions for this process are 30 degrees Celsius and a pH of 7. We will produce our PETACE and METACE extracellularly using recombinant E. coli. We will use sec-dependent signal peptides, as explored in some research publications, to produce cells with the ability to secrete PAM. We chose to produce our enzymes extracellularly because it significantly simplifies our purification process, as we only have to purify enzymes from the broth. It's also economically beneficial because we can reuse our E. coli. The production steps for this process are the same for PETACE and METACE. We will begin by obtaining our gene of interest and then use restriction enzymes and DNA ligase to splice together the plasmids and the gene. We will then transform the host cell using PET22B plus plasmids expressing our gene. We chose this plasmid because it has a relatively high copy number, high expression number, and has been used before for the production of these enzymes. It also carries the code for ampicillin resistance, which is useful for screening unsuccessfully transformed E. coli. After we screen and isolate the cells, we will then place them in a reactor to cultivate them and induce protein expression with isopropyl beta D1 thiogalactopyranoside. For the production of PAM, we chose to use a fed batch reactor because it's the best method for precise process variable control. It also allows us to maintain ideal conditions for cell growth as well as optimize the production of PAM. With fed batch reactors, we're also able to limit the accumulation of toxic byproducts such as acetate because we can restrict our glucose input. We designed a reactor to operate at 37 degrees Celsius because it's the ideal temperature for E. coli growth and also avoids PAM denaturation. For our inputs, we have glucose, which is our substrate, and E. coli at the host cell. And then our output are PETase, METase, and E. coli.
Along with our choice to use fed batch for PAM production, we made several important scaling decisions to size the bench, pilot, and manufacturing reactors. We based capacity on our second process, first determining the total amount of plastic we hope to degrade and using that to calculate the amount of enzyme we should produce. As we scaled up, we maintained geometric similarity with a 2:1 height to diameter ratio. We also used agitated reactors, which is fitting for our design since E. coli can withstand shear forces. In each scale, we determined the reactor volume and feed flow rate needed to produce a set amount of PAM with a cycle time under 1 day. For a long-term industrial goal, this results in 592 60,000 liter reactors fed at 30 liters per hour to produce about 1.5 kilograms per hour of PAM in each.
The next step is purifying PAM for use in degradation of PET plastics. We want to focus on process yield and productivity and not so much on purity because the enzymes are just going straight to the next manufacturing step. Because we are doing extracellular enzyme secretion, it simplifies the purification process a lot. It is more similar to CHO cell protein purification compared to regular E. coli. And because PETase and METase are very similar both in structure and chemical properties, the same purification steps can be followed for both. And these lab scale purification techniques can be directly scaled up to the pilot and then manufacturing scales. These purification steps are done or based largely on the work done by CO et al., who worked on producing these extracellular secreted enzymes. So we begin with doing a nuclease treatment and the removal of unwanted DNA/RNA, and then centrifuge and put the permeate through a depth filter to remove most of the unwanted cell debris. To further purify and filter our product, we will dialysize this against a buffer and finally do a column chromatography with the nickel NTA affinity agarose column. The tags on the enzyme should bind into the column and the pure protein can then be eluted out. We then want to crystallize our final form so that storage and transportation of our enzyme is easier for the next steps. The waste will be disposed of at appropriate facilities and the purified enzymes will be used for the next breaking down of PET.
Moving into phase 3, we will now be using PAM to break PET down into its constituent monomers. We chose to use a batch reactor for this phase due to two main benefits. The first is a shorter cycle time, which will allow us to optimize the amount of plastic we're able to degrade, and also it allows for a lower chance of contamination due to screening, traceability, and lack of a continuous feed input. One disadvantage is that cell contaminants are kept in the process longer. However, our purification process is fairly intensive, so we believe that will not be a problem for our process. We plan to hold this reactor at 30 degrees at a pH of 7, since that is the optimal condition for this reaction to occur. In terms of the inputs, we have two main inputs. The first is amorphous plastic, which will clean, heat to 255 degrees Celsius, and then cool down. And we also have PAM, which Michael mentioned in the last slide, but we will concentrate this to 0.75 liters of PAM solution per liter of water. In terms of outputs, we will see the plastic monomers, which we wish to further purify, BHET and MHET, which are the waste products, and the undegraded plastic, which is around 1% of the input plastic. This 1% is an assumption based on the kinetics of the enzyme itself.
Many of our scaling decisions here for plastic degradation are similar to those made for the PAM production process. We first sized our manufacturing scale based on our goal to degrade 10% of global plastic consumption and on the reaction kinetics, then scaled down to appropriate pilot and bench reactor sizes. We maintained the same 2:1 geometric similarity and again used agitated reactors to enhance plastic degradation. 592 200,000 liter batch reactors with a 23-day cycle time successfully degrade the approximately 2.5 million tons of plastic from our 10% global degradation goal.
Our next step is to purify our monomers for sale. We will begin by using microfiltration to remove any unreacted plastic from the aqueous solution. We will then use ion exchange chromatography to separate the PAM from the rest of the solution. We will then follow this using size exclusion chromatography in order to separate the ethylene glycol from the M het, B het, and terephthalic acid. We chose size exclusion chromatography because ethylene glycol has a much smaller molecular size than terephthalic acid, M het, and B het. However, we cannot effectively separate M het, B het, and terephthalic acid using this method due to their similar molecular sizes. Finally, we will use high temperature filtration at 200 degrees Celsius to recover the terephthalic acid from the M het and B het. We chose this method because M het and B het have a much lower melting point than terephthalic acid. For our equipment, we modeled the chromatography columns as packed columns and determined that we need a height of about 16 meters and a diameter of 4 meters in order to achieve sufficient separation. We will use a 100 square meter cast iron plate and frame filter for the high temperature filtration. In terms of storage and disposal, the B het and M het stream will be disposed of at local chemical waste facilities, and the monomers will be stored in plastic drums at room temperature before sale.
Jumping into the financials of our process, we wanted to discuss how long our company would need to become profitable and how exactly it would become profitable. So the first thing that we did was to determine the cost of our entire process. So we looked at both the capital and the operating cost, and we used the FACT method to determine both. The FACT method allowed us to calculate the cost for all the equipment used using the size dimensions, as well as the cost for any labor, utilities, and the inputs based on how much we would need for each of them. The costs for 1 reactor and 592 reactors are listed below for the Washington, D.C. plant, and then the eventual 10% of the total consumption as well. Next, we decided to calculate the revenue by taking a closer look at the plastic market in general. We found that the biggest consumers or producers of plastic were food and beverage companies that used plastic monomers for all of its product packaging. So we hope to partner with food and beverage companies, and we believe we will be able to do so since it's better PR for the company to buy from us rather than to source from oil and gas companies. The current market average of plastic monomers is 84 cents per pound. However, we believe that we might be able to sell at a premium since we are the first company to create a viable process for this. Even at these rates, we are able to have a very large profit of around $5.5 million for 1 reactor and $3.5 billion for the 592 reactors. While these numbers are very promising, the true success of a project is determined by the ROI, which we've now calculated in this slide. And as you can see, the ROI numbers are very promising. The ROI does increase slightly for the larger scaled up system, and this is due to any synergies that may occur when we are scaling up. Also, we calculated the NPV, which shows that the break-even period is less than 1 year, since even at the 1 year mark, the NPV is a positive number. Based on this economic analysis, we believe that our process will do very well financially and is economically feasible.
We also created a business development timeline based on this economic analysis. Before official product launch, we will validate our process works on the bench scale. Then in the first 2 years, scale up in size to pilot and manufacturing. We will implement our startup goal of operating with 75% of the plastic consumed in DC in years 2 and 3. Based on our financial calculations, we gain enough profit by operating a single manufacturing scale system for 1 year to build 4 additional reactor systems. So in year 4, we begin to scale up in quantity and continue this through year 12 until all 592 industrial systems have been built and begun operation to recycle plastic throughout the world.
Considering intellectual property a little bit, so our process certainly has utility on a global scale, and it's novel in scale and sequence. And what that effectively means is that there is nothing that currently exists that is performing the same function at this scale. However, the principles that we're using, for example, leveraging E. coli to produce these enzymes or using these enzymes to degrade plastic, these principles aren't novel on their own. So we might have trouble patenting this process at first because it might not seem non-obvious to people skilled in the art. However, in the future, as we develop our process, as we develop more parameters or specifications, we may be able to patent the process in the future.
In terms of risks and next steps, we first want to pursue appropriate environmental regulatory action in order to ensure that our process is doing more good than harm in the end of the day. We want to identify partners who'd be willing to invest in and purchase our monomers, companies such as Coca-Cola or Pepsi that produce a lot of these PET plastic bottles. We want to expand our manufacturing from DC to other cities around the world. And we want to improve the efficiency of PAM in our processes. And this could be stuff like recycling M hat and B hat back in order to maximize our yields. Some of the risks we identified would be no potential buyers. However, we believe that the sustainable environmental push will push companies to buy our product. And another factor could be contamination ruining batches, but we'd want to ensure that our plastic is clean and the heating process would kill any other bacteria. And thirdly, competition from traditional recycling methods. However, we believe that our method will still be more sustainable in the long run. So thank you for listening, and special thanks to Professor Delisa, Walternus, and Tina for all your help this semester.